We call this microscope the light sheet microscope for simultaneous multi-view imaging, and we developed and built this system to be able to image and follow fast dynamic processes in large biological specimens. In particular, we used it to follow the neuronal activity in an entire brain. Now, the way this microscope works is that we send thin sheets of laser light into the sample chamber, and the zebrafish would be located inside this chamber here in the center. And the sheets of light that we create here in these two illumination arms enter the chamber and illuminate the sample in a thin section, and then we image that part of the brain, record it with two detection arms here at a right angle to these illumination arms. You can see these fastest CMOS cameras at the end of each of these arms, one here, one on the other side. So by quickly illuminating the sample with these laser sheets and then recording images, we can very rapidly image the entire brain. And this is in fact the first time that an entire brain, an entire vertebrate brain, has been imaged at a speed that allows us to follow the neuronal activity basically in the majority of the neurons that the brain is composed of. In our study, we used zebrafish as a model system. You can see those here in the background. Zebrafish are vertebrates, and the hope is by using such a model system, we can gain insights from our whole brain functional imaging study that may potentially lead to insights about brain function as it applies to us as humans. So you can basically see the entire brain from three different perspectives. This is the top view, this is the view from the front, and this is the view from the side. You can see the different brain areas, the forebrain here in the top. This is the optic tectum, the hindbrain, all the way down to the spinal cord over here. So the red flashes that come up from time to time, particularly this big one just now, show the neuronal activity that we record in this fast imaging experiment. There's a structure that we found here that we call the hindbrain oscillator, where there are two groups of cells basically alternating. They're never quite on at the same time, so sort of in perfect antiphase. There's also a pattern here in the spinal cord that is linked to the activity that we see here in the hindbrain. And overall, these are fairly complex patterns, and this is exactly one of the reasons why we want to do this whole brain imaging experiment. We don't really quite know what to expect. I mean, nobody has seen really the activity in the entire brain at the same time, at these time scales and over this long period of time. There's a lot of potential in the capabilities that such an instrument provides. And I think there was something in the matrix.
The next step is to combine this technology with technology that I developed last year that consists of a virtual reality setup for zebrafish, so that they can actually move around in a virtual reality world and behave. So we plan to combine this, to combine these two technologies, and we'll keep collaborating on that. The other next step is to increase the temporal resolution, so to be able to scan the brain much faster than we currently do. This is interesting from the point of view of the Brain Initiative, because a big part of that project consists of observing neural activity in every neuron in a brain, eventually, hopefully, big brains, such as mice or human brains. But I think this is a step towards achieving that, since we're able to look at activity in all the neurons of a fish brain, which is also vertebrate and has similar brain structure to other vertebrates like mice and humans. I think the most important aspect is that it is now possible to observe what large populations of neurons across many different brain areas are doing. And this will allow us to make conclusions about how many neurons across different brain areas work together to achieve what the brain does.